Hello, Dr. Chen speaking. Hi.
Wei?
Hello. Hello. Hi, can you hear me? Hi, Dr.
Uh,
Chen.
uh, I think it's we have 3 minutes left.
Okay, no problem.
Okay.
Hi, Stella, how are you?
Fine. I have something to discuss with you about the money of this Congress, but I think we can talk privately.
Sure,
Uh,
sure.
I,
Okay,
I will send you an email to you and Vincent.
sure. Thank you very much, Stella. I know it's difficult, don't worry.
Um,
Remove from stage.
Time is up. Okay. The talk will begin right now. I will introduce today's speaker, Dr. Christopher Chen. He's a senior clinician-scientist and director of the Memory Aging and Cognition Center at Singapore University. His earlier medical and research training was at Cambridge, Oxford, and the Institute of Neurology, London. Since returning to Singapore, he has developed a multidisciplinary research programs with extensive international collaboration. His major interests are neuroimaging, molecular biology, and the treatment of stroke and dementia. He is a principal investigator for several large trials, served on editorial board and participants in trial steering committee, advisory panels, and consortia. Currently, Dr. Chen is president of Asian Society Against Dementia, ASAD. We just have a conference at Taiwan. Secretary Treasury of the Asia and the Oceania Association of Neurology, AOCN, and the past chair of the International Society for Vascular Behavior and Cognitive Disorder. He's also a co-chair of the World Stroke Organization's Future Leaders Program. Let's welcome Dr. Chen.
Thank you so much, Stella. Can I check whether you can see my screen?
Yeah.
Excellent. Okay. So really a great pleasure to go and sort of join you again and to go and sort of discuss how we can work together to develop the next generation of clinical care for dementia and what we at the Memory, Aging and Cognition Center are trying to do to advance the agenda. Of course, you know, all of us would rather be able to travel to Taiwan than to do this by Zoom. So I hope that the next time we'll be able to meet. And I'm particularly keen to sort of meet with colleagues and to see the facilities and to see what work we can do together. So what is this Memory, Aging and Cognition Center which I lead? Well, we are a collaboration between my university hospital, and various other agencies in Singapore and institutions in Singapore, as well as internationally. We have 4 themes which focus on what we think is, you know, particularly Asian, but actually global phenotype of dementia and cognitive impairment due to a combination of neurodegeneration and cerebrovascular disease. Today, we're going to talk mainly about Alzheimer's disease. And these themes include discovery of biomarkers and investigation of mechanisms, which is led by my colleague Mitchell Lai, a neuroimaging center where we have both MRI, PET, as well as retinal imaging, a clinical theme, which you hear more about, where we have both subjects from the clinic as well as from the community, and a epidemiological theme. And we've been very grateful for consistent funding over the last 2 decades now from the National Medical Research Council of Singapore. So the vexing problem that we all face is how we're going to deal with this anticipated large increase in dementia burden in Asia. Specifically, how are we going to prevent and treat cognitive impairment and dementia? Well, I think that it's time for us really to have a next generation clinical care pathway. We've been working to build up our clinical pathways over the last 2 decades, and I think that we all know that this needs to change, especially with the advances in treatment and diagnosis for Alzheimer's disease. So here's a paper, if you're interested, where I'm going to go and sort of use as a template, because together with other colleagues from around the world, I think we have described what we think might be a very useful way forward. So in summary, what we think that we should be doing is to have a wide spectrum of changes, beginning, of course, in the asymptomatic population in the community, moving on to symptom detection in primary care, as well as clinical assessment and differential diagnoses in the memory clinics, which will really depend now a lot on biomarkers to confirm the diagnosis, to advocate for certain types of targeted treatments, and then, of course, to monitor the effects of this treatment. And of course, we do have a number of tests available for clinical use now, but we also know that many of these tests are difficult to access and may not be implemented widely. And we also, and this is what we'll be talking about, have various tests under development for clinical use in the future, which I think will really revolutionize how we diagnose, care for, treat patients with Alzheimer's disease. So to begin with, we're going to talk about what we know about the beginning of Alzheimer's disease. I think that over the years now, we know that accumulation of Alzheimer's disease pathology in the brain is a very slow process, and that the transition from, the transition from healthy aging to early symptoms of dementia takes a long time. And this gives us an opportunity to prevent the accumulation of pathology and to build up resilience. And of course, we also know that there are various things that we might be able to do, such as exercise, social interaction, diet, and medications for risk factors. But this is just the recipe. How do we actually put it into action? And this is where I think that we need more clinical trials. So what we are doing in the Memory, Aging, and Cognition Center is that we've initiated a trial called the SINGER trial, which is based on the very famous Finnish finger trial. And we aim to recruit 1,200 community-dwelling elderly at risk of dementia to put them through a 2-year lifestyle intervention program. And we aim to add on biomarkers to investigate investigations. And we're trying to develop scalable digital platforms, build partnerships, and take this forward so that we can actually implement this widely across Singapore and perhaps Asia. And the two types of interventions we are trialing are either a self-guided lifestyle management, where subjects will be given tools to manage their own lifestyle changes, and a more intensive structured lifestyle intervention, where subjects will be given a lot more help to exercise, to diet, to train their brain, and to monitor their vascular risk factors. And this is, of course, part of our Memory, Aging, and Cognition Center's set of clinical trials. So in addition to SINGER, we've also participated in various investigator-initiated clinical trials, as well as commercially sponsored clinical trials. And we're able to do this because, like you, we have a good clinical research core, which specializes in clinical and neuropsychological assessments, as well as access to neuroimaging, biomarkers, and of course, other research imaging such as retinal and cardiac markers. Of course, what we hope to build, and I think that this is something we need to talk about with Taiwan, is whether we can build research consortia like the ones I've listed below. So we were able to get funding from the National Medical Research Council to conduct this trial. And of course, this would not be possible without the aid of a large number of colleagues in Singapore and abroad. So we have a group of epidemiologists and clinicians who are going to run the trial. We also have a group of neuroscientists and clinician-scientists who are going to help us do the brain, retinal, and blood biomarkers within this trial. And we have health economists to go and sort of link these results to public and clinical policy development. One of the most important things about this trial is how we have reached out into the community to provide lifestyle interventions. So one such initiative was that we linked up with a government, Singapore government agency called Active SG. Active SG runs a series of 12 gyms across the country. These are well-equipped gyms which are open for access to the public, of course, not during the pandemic, but then we've now reopened. And anybody can turn up, especially the elderly. And we've also linked up with the supplier of these gym equipment called HUR, who provide equipment which is very modern in that it can be digitized. Each subject has their own card which they carry around with them, which knows what kind of exercises they need to do, what level they've reached, and it can be individualized. We've also, of course, in this time of COVID, been able to provide physical interventions through home digital Zoom. So we can educate our subjects on the types of exercises, we can demonstrate, we can actually correct posture, and then we can poll people to see how well they've done. And of course, we can keep track of whether people have been exercising. In terms of cognitive interventions, we've been able to give online group talks as well as discussions and interactive activities. So again, we don't have to do face-to-face meetings anymore. We can do all this virtually. And in terms of online material, we have developed a lot of material which teaches patients about diet, cognition, memory, and exercise, which I think that they can all refer to because we provided all our subjects with tablets. And finally, moving back to the biomarkers, this study also will have, I think, two-thirds of the subjects will have MRI scans. Everybody, hopefully, will get retinal imaging as well as blood biomarkers. And I think that this would be a very good way of trying to discover whether we have biomarkers for people at risk of further cognitive decline, as well as biomarkers for response to intervention. And of course, biomarkers predictive of treatment response. And one of the key lessons that we've learned is how important it is to partner with existing organizations within the community. And thankfully, Singapore has several of these community organizations, in particular, the Lions Befrienders groups, which are well integrated in the community, have the trust of the elderly in the community, and are able to intermediate between those of us who come from the university, these ivory towers, and the actual community. So we're very grateful to them for publicizing our study, for introducing us to their clients, and also in supporting our initiatives. Finally, I'd like to go and sort of mention that we are part of a very large population health initiative by my university. And in fact, we have embarked on a unique collaboration with various government agencies, including Housing Development Board, the National Parks Board, and academia to develop a health district at Queenstown, which is one of our older estates in Singapore. We selected Queenstown not just because it was close to the university, but also because its demographics very closely mirrors Singapore's projected national demographics, which is an aging population. And we're going to implement, together with our hospital and health system, programs to improve the residents' access to preventive health services, to bring in health screening on site to residents. And as you hear later, we want to initiate cognitive tests to be able to detect early cases of cognitive impairment. And of course, these subjects are invited to join the Singer study. And so far, it's going quite well. We've recruited almost 400 subjects in a year, and many of our subjects are very happy with the interventions that have been performed, and 2 of our subjects have been featured in the newspapers, and so we hope to be able to complete this trial within the next couple of years. Next, of course, apart from preventing cognitive impairment and dementia in the community, what about early symptom detection, better clinical assessment, and differential diagnoses? So currently, we try and use cognitive screening tests. We do standard examinations. And in future, of course, we hope that using blood biomarkers, digital technologies may improve our ability to perform these important functions. But, you know, there are lots of drawbacks at the moment. And one of the problems, of course, is that our primary care colleagues may have a lot of difficulty in detecting the earliest subtle changes in cognition and behavior. We also, of course, need to educate our population to proactively seek clinical assessment if they feel that there are early symptoms. And maybe opportunistic detection during routine visits, not just of cognitive impairment, but also perhaps of biomarkers which may indicate that you have a higher risk of dementia, would be the way forward. The standard medical assessment sometimes takes too much time, too much effort, and maybe we need to go and start relying on the use of digital assessments and blood biomarkers. So in the paper that we wrote, I think we've set up a sort of pyramid of things that can be done and how digital technology can help. Of course, digital technology has many strengths. It's convenient, it's accessible, you can generate a lot of data continuously. You can use AI to detect, look out for patterns. And of course, it's very individualized and patient-centric. But of course, these are also drawbacks with things like privacy and cost. And the clinical utility of such digital tools is, you know, obvious and increasingly appreciated. We can use digital tools to screen and detect early. We can, of course, especially in a pandemic era, avoid face-to-face meetings and do remote assessment and remote monitoring. And of course, this may be more convenient and cheaper as well. And digital tools may improve clinical patient engagement, adherence, and obtain more caregiver support. But you know, there is a hierarchy of digital health data. The traditional way of collecting data has a very high burden, often has to be done in clinic and relies on traditional health data. More modern assessments have lower burden, but you still need to go and sort of get people to go and sort of do things. And I think that the future may lie in passive tools which just automatically collect data and analyze it, such as collecting your sleep patterns, your mobility patterns, and testing your hearing and vision. What we have decided to do in Singapore is a much simpler study. This is called the DECIDE study, where we are using digital tools to detect cognitive impairment and dementia in the community. And we're doing this because we realize that even though primary care is very important for MCI detection, our colleagues, the primary healthcare providers, often lack technical support, infrastructure, training, experience to detect and manage such patients. And in fact, many studies have shown that primary care health providers lack confidence in making diagnoses on these kind of cognitive states. And they also they have uncertainty around what cognitive assessments to deploy, how to perform the cognitive assessment, and importantly, of course, what to do after you assess cognition. We realized that actually there's a lack of perspective from patients and service users on the effects of early screening and the implementation of such efforts. Furthermore, COVID-19 has really impacted all our previous efforts on case finding and diagnosis because patients are reluctant to come to clinic and reluctant to have face-to-face assessments. So the gaps that we're going to address by this project include determining whether we can have accessible screening tests for the community, whether we can come up with a better system of referral, and getting to hear what community members really want. So we want to implement and evaluate an early screening system to detect cognitive impairment and dementia in the community. And what we're going to use is a digitized version of the quick dementia rating system, which some of you may be familiar with. This questionnaire, it has only 10 questions, takes only about 2 or 3 minutes to do, and has validated cutoffs, which can indicate whether you have no memory problems, some mild memory problems, or moderate to severe cognitive impairment. And we have devised a website where we can do these tests online and give people advice online as well. Now, the idea is that this test, which is highly sensitive in recognizing both MCI and dementia, and has very good correlations with traditional cognitive assessments and measures, can be administered online. We have already validated it in the clinical population, but it needs to be validated in the general population. And these are just 2 recent papers, one from ourselves in Singapore and one from South Korea. The first phase is to validate the QDRS in the general population. And we are going to go and sort of recruit from various channels, from senior centers, like we talked about when we're doing our clinical trial, health fairs, as you can see, that our institution organizes and are very popular with the community, educational talks, and advertisements. And we're going to ask people to assess themselves. And after they've been assessed, we want to invite them back for full assessment at our research clinics, and then to see whether we can validate their QDRS. Now, of course, that can't be the only thing we do. So in parallel, we're going to do some formative research to decide on acceptable and efficient referral pathways. So we're going to get community partners, residents, and our community health posts to come and form groups to discuss what's the best way of dealing with referrals, to synthesize the evidence, to confirm it with stakeholders, and to go and produce a viable pathway. Finally, of course, we are going to try and implement this in the community and see whether this really works and whether we can sift people in the community health posts, which are our first line of healthcare in the community, between those who really need to see a memory clinic, those who may have predominantly mood and behavioral problems, which need to see our psychiatrist, or those with multiple morbidities, which need to be sorted out first, either in the primary care or through the geriatric clinic before a final diagnosis is made. Of course, we wish to assess whether this process has actually worked. And finally, we now come to what we're going to do in a memory clinic setting, in a specialist setting, how we perhaps can use biomarkers to diagnose, to initiate treatment, to monitor treatment, etc. And I think that you will find that although we do have established biomarkers for Alzheimer's, I'm going to emphasize blood biomarkers. And I apologize if you're going to hear this again, because I did speak about it before at the ASAT meeting. And I think you will hear from Professor Kim again in your next talk about blood biomarkers. But really, this is very important for our work in the future. So in the second line, in the AD Specialist Center, diagnostic workup and therapeutic decision-making will be based on a comprehensive assessment, neuroimaging, as well as the new ATN-X classification. And this table just summarizes what we mean by that, that in future for Alzheimer's disease, we're going to be able to use biomarkers to decide whether people have significant amyloid load, significant tau, neurodegeneration, and perhaps other markers of pathophysiology, such as neuroinflammation, so that we can give targeted treatment. So of course, you know that right now we only really have targeted treatment for amyloid, but targeted treatment for tau and for neuroinflammation is coming along. And I think that combination therapy is probably the way forward for Alzheimer's disease, just as it is for oncology and other complex diseases. So biomarkers, I think that you're all familiar with this, can be diagnostic. In other words, they can tell you whether somebody's got a disease or not. They can be prognostic, means that they can predict what's going to happen to you. Having a particular level of biomarker means that you're likely to progress, convert to dementia, or perhaps if you have low levels, maybe stable. They must be able to predict response to interventions when, of course, we have interventions, and hopefully in future, we use as surrogate endpoints for clinical trials. Currently, and previously or currently, our range of biomarkers in Alzheimer's has been really restricted to clinical exams, neuropsychology, some forms of neuroimaging, and too late, usually, neuropathology. But the previous clinical criteria for Alzheimer's disease really didn't use all the advances that we understand about the biological basis of Alzheimer's disease. So not surprisingly, the diagnosis of Alzheimer's disease or the criteria for Alzheimer's disease has changed over the years. This is just a summary of how it's changed since at least I started doing Alzheimer's disease. Namely, the main change has been moved from a clinical set of diagnoses into a biomarker-driven diagnostic pathway. And of course, I'll quickly run through this. This was triggered off by research criteria for Alzheimer's disease by Bruno Dubois and others. And they proposed using MRI. All of us are familiar with cortical atrophy, and particular hippocampal atrophy, metabolic patterns of disease such as FDG PET, but more importantly, sort of molecular neuroimaging methods such as amyloid PET to show pathology in vivo. And this has led to a change in how we diagnose Alzheimer's disease. So it's still possible to diagnose Alzheimer's disease using just clinical criteria, but we all realize that this means that about 30% of our patients may not actually have Alzheimer's disease pathology. And so to improve this, biomarker have been brought in to increase the probability of the diagnosis and also to rule out Alzheimer's disease. And not only for Alzheimer's dementia, but also MCI due to Alzheimer's disease, and increasingly the preclinical stages of Alzheimer's disease. So I think we're all very familiar with the Jack curve, where we understand that amyloid may start accumulating very early in the disease, but that neurodegeneration and cognitive symptoms follow much later. But nevertheless, in the preclinical and early dementia cases, we have an opportunity to prevent neurodegeneration and cognitive deterioration. So I won't bore you with the ATN framework, but I think most of you know that if you have Alzheimer amyloid, you become part of the Alzheimer continuum. And if you don't have amyloid, you're either normal or you have a non-Alzheimer pathological change, especially if you have neurodegeneration. Currently, our core biomarkers are PET, MRI, and CSF. But I think all of us know that there are issues with all these biomarkers in terms of expense and accessibility and invasiveness. So less invasive blood biomarkers may be crucial in the future. And if you're interested, my group has published a very nice review on recent advances on the various ways that we can detect amyloid beta as well as tau. I think it's important to emphasize that really a lot of progress has been made in the last decade to give us really highly sensitive assays. And also moves have been taken to measure specific isoforms of either A beta or tau, which may be more relevant to Alzheimer's pathology. So the most sensitive amyloid detecting technology, unfortunately, is also the most expensive, which is mass spectrometry. But I think that we all realize that more accessible tools such as Simoa or IMR will make this more practical for use in our clinics all over the world. And of course, there are recent advances in very specific methods to detect oligomers, which are promising, but have yet to be fully validated or optimized. Now, there are increasing numbers of head-to-head comparisons of these various blood biomarkers, and I think it's fair to say that these blood biomarkers actually perform reasonably well, and that they can really sort of allow us to pre-screen patients before they go on to more either invasive or expensive confirmatory tests. The same can be said for phosphorylated tau. And again, as you can see here, there are many different kinds of tau species that we can look at, including phosphorylated tau 181, 217, and 231. Suffice to say, the most studies that have been done are on p-tau 181. And I think it's a challenge to decide which assay to use, because what appears to be most sensitive may also be the least accessible set of tests. Nevertheless, I think that there is promise in combining these tests. So instead of just using p-tau or A-beta 42, combining them as a ratio may turn out to be a much more sensitive way of determining who has got increased amyloid burden. So in this paper that we published last year, the p-tau A-beta 42 ratio turned out to be the most sensitive in distinguishing patients with amyloid burden. And we are now setting up a consortium, a worldwide consortium, to see whether these results are widely applicable. Moreover, these tools are becoming recognized for use. So the US FDA has recently given a breakthrough device designation for p-tau 181. Of course, other tests have also been given such breakthrough designation. But the reason we're so excited about this is that we're going to start using p-tau 181 for further studies, which I'll describe in a moment. So right now, our biomarkers for dementia are moving very quickly. We have neuropsychology, neuroimaging, CSF, and emerging blood biomarkers. Blood biomarkers, however, need to be brought to the clinic. I think that this wonderful paper by Charlotte Teunissen summarizes how we have gone through phase 1 to phase 3. In that preclinical studies, clinical assays, retrospective and longitudinal studies in research setting, but now we really need to do prospective studies and real-world performance so that we can implement in to the clinic. So the future really lies in biomarkers, which are what we call first-line optimal biomarkers to rule out, to diagnose, and then to allow people to go and sort of access other diagnostic tools. And so we need to go and sort of work very hard to bring biomarkers into clinical practice. So what we have proposed to do in the MACC is something called the Early Diagnosis of Dementia Study, the EDEN study, where we're going to recruit subjects from the memory clinic, do blood biomarkers on them, and provide these blood biomarker results to the clinicians to investigate whether knowledge of blood biomarker results will change a composite endpoint, which includes diagnosis, change of medication, change of management. And I think we are very confident that this will be successful. And this will allow us to go and sort of do these very important prospective studies and help to implement blood biomarkers, not just in the memory clinic, but perhaps in primary care as well as in population screening. And that's because these blood biomarkers are so cheap. And if they're cheap and effective, I think it will transform how we manage care and allow us to really pick up early symptoms of signs and not ignore them and not miss the boat. And why is it important not to miss the boat? Well, I think that it really is because we now have the hope of targeted treatment. I think all of us were excited when aducanumab was reported as being able to eliminate amyloid in amyloid-positive patients. But we were all disappointed when, when the trial results came out and showed not very convincing clinical trial results. Even though it eliminated amyloid, it wasn't able to show a huge change in the clinical outcomes. And this is the positive trial, because the companion trial was negative. So I think that most of us have been through this terrible saga of how the launch of aducanumab, the first approved anti-amyloid disease-modifying treatment, was approved last year, and aducanumab lasted longer than the current British Prime Minister, but you know that it was very controversial. Clinical evidence was thought to be too complex and not sufficiently convincing. It wasn't accepted in the European Union. The Biogen pulled out from Europe, and the death knell came when both the CMS and Medicare refused to agree for reimbursement in the US. So this has led to Biogen retrenching a lot of its staff and basically killing off aducanumab. But there's still hope, because I think you know that other companies are still pursuing anti-amyloid treatments, and this is Roche's gantenerumab drug, where they have now launched into, have completed a phase 3 study using a much higher dose than they did before, which really does eliminate amyloid, and the results are due very soon in this quarter. And I think that we are also familiar with the ICEYE phase 2 trial of lecanemab, which showed that this drug, which antibody which targets amyloid fibrils, does reduce amyloid, and the optimal dose, which was brought forward into the phase 3 study, does seem to have a clinical impact. And we all thought the results would be due. Not everybody was sure what was going to happen, and last month, this fantastic press release came out that showed that the lecanemab was positive. Not only did it have meet its primary endpoint, which was reduction of clinical decline as measured by the clinical CDR sum of boxes, but all other key secondary endpoints were also met. For the younger ones, this is amazing, because we haven't had such a success in amyloid in Alzheimer treatment in decades. So the results will be published soon and will be discussed at the next CTAD meeting. So this is a really exciting time for dementia. We have new treatments coming on, and there's really an imperative to change our clinical care pathway for the future, not just for our patients and the families, but also for our societies. Thank you very much.
Thank you, Chris. Any questions from the audience? Okay. Weiru, you can talk directly.
Okay. And Dr. Chen, Dr. Lee from Taichung. And
Hi.
thank you for your talk. And my question is there any difficulty when you asking the study participants to have multiple times of different kind of PET scan because of the radiation? So some of our participants are afraid of the multiple radiation exposure. Thank you.
Thanks, Dr. Lee. Are you referring to doing both amyloid PET and tau PET?
Yes, even the neuroinflammation PET.
Okay. Well, I suppose, okay, so the way I look at it is that we have radiation guidelines. So the FDA allows a maximum of 50 millisieverts per year for research. And each of your PET scans is about 7 or 8 millisieverts. So you will need to do a lot of PET scans to reach that FDA safety margin. Now, in Singapore, for a number of years, we had a radiation safety limit of 10 millisieverts, which did make our lives very difficult. I mean, but so we actually had to do a number of very strange things, like the rules said that you could give a maximum of 10 millisieverts per calendar year. So we would try and do all our amyloid PET scans in October, November, December, and all our tau PET scans in January, February, and March.
Oh, okay.
But that's very silly. I mean, so it really doesn't make sense. So what we have done is to encourage our authorities to recognize that this is really too low a limit, and that we needed to go and change the limits to reflect the the significance of the research and the seriousness of the disease. Now, that's for the authorities. For the patients, what we explain to them is that this exposure is really still quite low, because the exposure you get, just by living, especially if you come from a granite area, the background radioactivity is quite high. It's already several millisieverts per year. And in fact, every time you take a flight, you're exposed to a lot of radioactivity. And the radioactivity that we give to patients using PET is a hell of a lot less than if you do multiple X-rays or CT scans, for example. So I think if you explain it to people in a rational way, most people will be happy and accept it. Of course, some people won't.
Yeah.
I don't think you can persuade everybody. All I can say is this, okay, we have, for the last decade, desperately tried to offer people CSF for lumbar punctures, for CSF examination. We've managed to persuade nobody. I don't know, maybe I'm very bad at it or something like that, but our research participants have said, thank you, no. But when we offered them PET, they all agreed. So these things are balanced. So I don't know. I mean, maybe Taiwanese are more risk adverse. I'm not sure. Of course, in terms of clinical studies, at the end of the day, I think that we will only really be doing amyloid PET if it's needed. But that's only one exposure. And if people don't want radiation, you can tell, okay, we'll do CSF. And then I think they will jump for the PET.
Okay, thank you. Yes, I mean, yes, I think I find a way to convince our patients to do the exam. And sometimes I also explain that when experiencing the flight to United States and go back to Taiwan, and the radiation is maybe much heavier than do a PET. Yeah.
I think the mountains in Taiwan are granite, right? So even walking, taking a hike exposes you to radioactivity.
Yeah, thank you.
Thank you, Dr. Lee.
Any other question? Okay, and Chris, how many subjects do you recruit in a single study?
Oh, we aim for 1,200, and so far we reached 400. So we are 1/3 of our way there. But I think that we had a lot of problems initially because we were foolish enough to think that we could recruit from our clinics, etc. But now we've learned our lesson, and we have built these partnerships with the community, and recruitment is really quite good. We are recruiting about 70 patients, 70 subjects per month now. So we should reach our target before the end of 2023.
And do you have a problem to keep the subjects in the program?
Well, so far, not too bad. I think that one of the key things is how enthusiastic and charming our team is. They are able to persuade our elderly subjects to stay on track. And I think the other important factor is that we don't recruit people. Of course, we recruit everybody individually, but we put people into groups. So they all do these activities in groups of 10 to 12. So there is, some comradeship and some peer assistance to stay on the program. But of course, there will be people who drop out for a variety of reasons. But I think it's very important to have good coordinators and social interaction.
Mm-hmm. Another question is that for ATN, and what kind of tau pad you used in Singapore?
Thank you. Well, we use the servo tau pad, but again, we are only using it for research purposes because it's not commercially licensed, and we haven't been able to get the other tau pad ligands into Singapore because it's getting PET ligands in is really not easy. It's a lot of complicated commercial and legal negotiations. Maybe you are better at it than we are.
And is it available amyloid PET in clinical service in Singapore?
Not yet. Again, okay, well, let me take that back. So commercially, the GE compound is available, but it's ferociously expensive. I think it's about 5,000 Singapore dollars a go, and I think that's partly because the demand is so low at this moment in time. And as you know, a lot of the cost of PET comes from the synthesis. And if you're only sort of synthesizing it for one person to use, then all that cost is shared by that one person. It goes to that one person. But I think as demand goes up, and especially for F18 compounds, where you do one synthesis cycle, you may be able to scan maybe 5-6 patients at a go. Cost will hopefully go down. So that's the GE compound is available commercially. For research, again, we use a servo compound, which I think is much better than the GE compound. Again, that's only for research right now.
So the amyloid PET in Singapore is for research purpose most of the time. Most of the time for research purposes, but very occasionally, and this usually happens in the private sector, commercially available PET, amyloid PET is performed. Okay. Yeah, but we really haven't reached a stage where, well, of course, things may change very soon with the projected licensing of disease-modifying drugs, but we don't know what the labeling requirements will be. As you know, the initial labeling requirements for aducanumab did not specify the use of amyloid PET. The FDA initially said that you could, give it to anybody who had a diagnosis of Alzheimer's disease. Of course, there was a huge uproar, and Biogen pulled back and said, well, you need a biomarker evidence. So depending on what Eisai and other companies agree to, perhaps there will be a need for more amyloid PET. I mean, so for example, in Taiwan, do you use amyloid PET, and what do you use it for? Similar in Singapore. We have available amyloid PET approved by TFDA, but most of the time we don't use it in the clinical practice. I see. Okay. Because it is expensive. Yeah. Yes, it is. So we have to find ways
of reducing the cost. And I think that the best thing to do is to try and pool together the people coming for scans.
Yeah. And Dr. Huang has a question. How many kinds of digital data are gathered? Is language included? Are wearable device used?
Thank you very much for asking an important question. So right now, as you can see, in our study, we're only doing this digitized questionnaire. Now, of course, I know a lot of people are interested in digital data, which gathers a lot more information, including wearables. We don't think is ready for prime time yet in the community. We are including variables into the SINGER study to see whether we can, the acceptability and also what kind of data can be used to see whether people are compliant or have changed their lifestyles, etc. I like this question about language. I know that, for example, one of my colleagues in China, who used to be our research fellow, presented at ASAD a voice recognition program where you could do cognitive tests online. Unfortunately, we can't use that in Singapore because the dialect was different. She did her studies in Hangzhou. And in Singapore, there are lots of different languages. And even for the Chinese in Singapore, there are lots of different dialects used. So, of course, I think all of us may have recently heard that Facebook has developed artificial intelligence method of understanding Minanhua, or we call it Hokkien in Singapore. So maybe we will try and use that to see whether we can, assess our patients automatically online. The other way of using language tools, of course, is to see whether you can analyze, the richness of language, the grammar, the various cadences, etc., to go and pick up early symptoms. But no, we are not doing that. I know that many people are doing excellent work on this, and I look forward to seeing their results. So somebody has sent me a form. No, it's not a question.
No, but
Okay.
do you use wearable device?
Only in research.
Oh,
Okay. I personally, I don't use a wearable device. I already have my wife and my daughter scolding me and telling me to exercise more. So I think that they are my wearable devices.
So for the digital data that you collect gait.
Yeah. So the wearable device that we are going to test out is the Aura Ring. So the Aura Ring really only detects activity and sleep, and it can also detect heart rate and temperature. But I know that people are picking up, looking at all kinds of things. I don't know whether it will really be helpful yet, because I haven't seen any convincing work showing that, firstly, can you cope with all this huge amount of data? Can you, in a reasonable time, analyze it to pick up patterns? And do these patterns actually have any clinical value? So until we sort these things out, I don't think it's ready for clinical practice yet. It's just like wearables for cardiology. We've had wearables which can pick up heart rate for some time now. But even so, none of them have really gone into clinical practice because they still have a high error rate. I know that Apple says that, the Apple Watch is not a clinical device.
Okay. Yeah.
Then I don't know about you in Taiwan, but we have a lot of complications with privacy rules in Singapore. the more data you collect, the more problematic it is to know whether you are collecting too much.
But the way about digital data in dementia study, it is a hot topic.
Yes, indeed. And I know that your center, at least the TMU center, is participating with the DAC on various digital tools. So I mean, I look forward to seeing the results. I think they will be wonderful. We are not so sophisticated in Singapore. We just want to try this digitized questionnaire first. If our subjects can't even handle that, I think it's going to be very difficult to do more.
Mm-hmm. Okay. If we don't have further question, then the talk will be end. And thank you, Dr. Chen, for such an excellent talk.
Thank you so much for inviting me. And of course, if anybody else has questions, please feel to contact me. And you're very welcome to come and visit. Please let me know if you want to set up any collaborations. Thank you very much.
Okay, thank you.
Oh, yeah, definitely.
Yeah,
bye-bye.
Thank you. Bye-bye.
Bye-bye. Oh, by the way, can I just ask a quick question?
Okay.
Okay. Is there a Taiwanese collaboration for clinical trialists in dementia? So do you have a sort of central group that deals with clinical trials?
Yeah. Yes. We do have a dementia dementia clinical trial group.
Excellent.
But I'm not sure that if it's still working.
So I tell you what, Stella. I mean, I occasionally get inquiries about whether different parts of Asia are interested in novel clinical trials. So what I'll do is I'll recommend that they contact you.
The president of Taiwan Dementia Society.
Okay. President of the Taiwan. So what I'll do is that I'll ask you for the email. Is that okay?
Yeah. Yes. I would like to help.
Thank you so much.
Yeah. Okay.
Thank you. Thanks everybody.
Thank you. Bye-bye.
Bye-bye. Bye-bye.